Hello, everyone. Welcome to my presentation. I'm Torsten from Liverpool. Thank you for the invitation by B. Wild and Dr. Suarez, allowing me to make a contribution to the conservation medicine workshop initiated by them. Let me share my screen and start my presentation. I'm Torsten, Dr. Torsten Wronski, Senior Lecturer in Animal Behavior and Wildlife Conservation at Liverpool John Moores University. I was working for most of my time on ungulates, starting with my studies on impalas and the social organization of bushbok in East Africa, mainly in Uganda and Rwanda. And I worked for the Zoological Society of London at King Khalid Wildlife Research Center in Saudi Arabia, mainly on the captive breeding and reintroduction of endangered ungulate species, such as the Arabian oryx, the Arabian gazelle, or the Nubian ibex. In relation to that, I also studied their predators and parasites. Subsequently, I moved to China, working on the captive breeding of musk deer, the endangered deer species bred for the production of musk and for reintroduction into the wild. Many years I spent working on the terrestrial gastropod fauna and their community composition in the tropical rainforests of the Albertine Rift Valley in East and Central Africa. And I have carried out a survey on harbor purpose in the North Sea. I will give two presentations during this workshop. One in module 3 on wildlife reintroductions, and one today, term population estimation and census techniques. Today, we will have a look at different counting methods of larger wildlife species, such as drive counts, point counts, transect counts, whether aerial or ground, how to fix the strip widths, and eventually we will come to modern distance sampling and using specific softwares. In the second part of this presentation, I will give you some insights into camera trapping and into the recapture method. Okay, let's have a look. Usually, conservation managers try to, if they want to count a population, they try to include as much area as possible. We call that total count. But this is, to various reasons, not always possible. These reasons, these limitations include, for example, financial reasons, time constraints, limitations in the availability of staff, the season and weather conditions, but also the terrain and the geography of the study area. So total counts are not always possible. And that's why we scientists usually use a subsample, a sample count, which is representative of the total area. In this case here, we have a total area subdivided into 10 sections of equal size. And we choose 4 of them to carry out our sample counts. We establish transects within each sub-area and count the number accordingly. In the first one, we counted 14 animals, in the second one, 9, in the third one, 10, and in the last one, again, 9. Taking the mean of these counted numbers, we will get 10.5 animals per subsample. If we extrapolate that to the total study area, we need to multiply it by 10 sub-areas, and we will have an estimate of about 105 animals in our study area. These results were confirmed by a second study, and the results from 20 samples of 4 units from the same study population. And the result was 100.2 animals with the range from 87 to 123. This is very much in line with our estimated or extrapolated result of 105. So subsampling is not ideal, but results obtained from subsampling are usually relatively close to the true number of animals in the target area. Let's have now a look at the different counting methods usually applied. First, the drive count. Drive counts are carried out by a line of drivers. These are usually people walking in a line parallel to each other in a certain direction, and therefore driving the animals towards observers here at the fringe of the count area. Some animals try to escape, across the drive line. Others are driven towards the counters and past them. In either way, the counters or the drivers will count all passing animals. However, this is only possible for smaller protected areas. If we have larger areas, we can take again a subsample. In this case, counting strips, 4 counting strips, and each strip is counted or driven by 3 drivers, driving the animals towards counters, which are here in this case waiting for the animals in vehicles. The distance between the drivers should be not more than 75 meters. Animals which go reverse passing the driver line will be counted by the drivers. Animals going towards the counters will be counted by the counters. However, drive counts are difficult to organize. They are intensive in labor and often not suitable because of the terrain and the geography. Another example are the point counts. These are perhaps the simplest method of counting birds, in which a trained observer records all birds seen or heard from a point count station, which is usually in the center, for a given period, usually 3 to 5 minutes, in an area with a radius of 20 to 50 meters. A series of point counts is usually completed over a fixed route, such as a transect, and can then be compared to the results of the same point count in other seasons or years. So usually such circles, point counts are established either along a transect, here in this case 1000 meter, with an interval of 200 meter, or they can be established within a grid of squares, whereby the sampling point is in the center of each grid cell. More recently, there was a software developed, freely available in the internet, called Point Count 2.02, and is freely available under this link. Another commonly used method counting wildlife species are the road strip counts. Hereby, a vehicle or a pedestrian follows roads which are pre-established, either within the park or the protected area. This is usually done because many animals can be included. Also the smaller species. If you drive at a slow speed, you will see a lot of animals from the car, whether large ones like elephants or small ones like dik-diks. But there are several disadvantages connected to this method. First of all, the road system needs to be well established and should cover most parts of the study area equally. The road system is often biased towards certain habitat types because tourists want to see animals in free open country, and therefore the roads are often going through those habitats which are liked by the tourists rather than through dense vegetation or rocky terrain. Another disadvantage is, or another prerequisite is that an accurate measurement of the distance of each sighting from the road is needed. As we will learn in a minute, this applies not only to road strip counts, but also to transect counts. And the vehicle is needed, is often expensive. Therefore, higher costs are involved than during drive counts or point counts. And the analysis of such data, of road count data, is pretty complex due to the fact that we would have to subdivide the road transects, the road counts, into several parts due to changes in the vegetation. Interesting study who has applied this method is the recovery of ungulate populations in the post-Civil War Akagera National Park in Rwanda, whereby road counts were employed over several years to monitor the increasing wildlife population after the park was reduced in size in 1997. Now let's have a look at transect counts, the most famous and most used method of counting wildlife. They can be aerial, ground counts, or for vegetation, or even underwater. Important is that straight lines are established in parallel orientation to each other, and to each side of the counting line we will have a counting strip. The size of this counting strip varies. We will come to that in a minute. First of all, we need to think about the baseline and the orientation of the baseline. The baseline will be established in order to establish transect lines in a right angle to the baseline. Here we have a study area somewhere in Zimbabwe, and through the study area we have a river going. And as you can see, these dots here are animals, and they are somehow clustered along the river, most likely in the riverine vegetation. Now the question to you, is it better to have the baseline across the study area in a right angle to the orientation of the river, and therefore transect lines running parallel to the river? Or would it be more advisable to have the baseline oriented parallel to the river, and therefore the transect lines running in the right angle to the orientation of the river? In what case do you think we will get better count results? It is this example, whereby the transect lines are running across the area, sampling each transect sampling different habitats at an equal ratio. Here we will have one transect line during which we will see all the animals, and in only one vegetation type, while on these transect lines we will have hardly any animal. Okay, let's have a look at aerial transect surveys, usually carried out by a fixed wing aircraft, a fixed high wing aircraft, because if the wings would be low, they would obscure the free sights of the observers inside the plane. Now, in order to restrict the count area, the strip width, to a reasonable width, we would need to fix markers on the plane. These can be streamers fixed on the wing struts of the plane, leaving a space between them, which will be basically marking our view area. These marks can be also fixed on the windows. Now, why is this the case? Let's have first a look at this graphic here up right. This is the transect line. This is the turning, which is usually not included into the count here is the ending point, and parallel to that, the next transect line will be flown. Usually, they should be not less than 2.5 kilometers apart. And yes, you can see here now we have two viewing areas to the left and to the right of the count transect, marking the strip width on the ground. How can we establish this strip width? Here in this case, it is 150. In our sample calculation here, it will be 100. So we want to have a strip width whilst flying on the ground of about 100 meters. So how do we need to fix our streamers or our window marks in order to have a sample area when we fly a strip width of 100 meters to each side? Now, in the first place, we need to establish some variables. First of all, we need to measure the eye height of the observer when being on the ground. This is the small h in our formula. Secondly, we need to establish or determine prior to starting the flight our flight height. In this case, 90 meters above ground. And we know now that we want to have a strip width of 100 meters on both sides whilst flying. So using this formula, we will take 100 meters times the eye height of the observer on the ground, 2.57 meters, divided by the flight height, and that comes to 2.85 meters strip width when the plane is still on the ground. So the observer will sit in the plane, looking at the ground when being still on the ground, and a second person will fix now the streamers in the way that it covers exactly a strip width of 2.85 meters on the ground while this plane is standing. Now we know if we fix our streamers on the struts accordingly or on the window, and we fly at a height of 90 meters, we will have exactly a strip width of 100 meters to both sides. So what do we need to consider if we plan and implement an aerial transect count? First of all, let's have a look at the abiotic factors. Of course, we need to determine the flying speed, which should be as slow as safely possible. And we need to determine the flying height. In our previous example, 90 meters, it should be somewhere between 60 and 100 meters. The speed, as slow as somehow safely possible. And we need to think about the counting time in the morning, during the midday, or in the afternoon. And we need to think about how to record the data. Do we use a notebook? Do we have a third person recording the data? Or do we speak into a portable voice recorder? And we need to consider weather conditions, cloud cover, the angle of the sun, very important, so that we are not dazzled during the flight in either direction. The wind speed is very important, and the direction of the wind. Imagine the plane is driven around by the wind whilst being on the transect line. That will have an effect on the view area determined by our streamers here on the wing struts. If the plane is in that orientation, the sampled area on the ground will be reduced, while flying in this orientation, the sampled strip width will increase. However, exhaustion of the observer and fatigue are another important factor to be considered. So the counting time should be not too long, because the eyes get tired and animals might be overseen. And again, most important, the strip width. And we will have now in a minute a look at how to best establish the effective strip width. But let's first have a look at the abiotic factors, which need to be considered prior to the aerial transect count. These are the soil and ground cover, whether we have sand, gravel, or rocks. Do we have vegetation in our count area? And if yes, what? Do we have trees, shrubs, or only an open grassland? You can imagine that this has an impact on the species identification from above. In an open country like here, buffaloes can be easily distinguished and counted, the same in desert habitats like here with the Arabian oryx. But in areas with dense vegetation, shrubs, or a woodland, a species like a bushbok would be difficult to see from the air. So we need some training actually before we start our count, training on how certain animal species look from above. We all know how they look from lateral view, but if we see them from dorsally, they often have a completely different appearance. And we need to consider the size, the behavior, and the activity pattern of our target species. Some animals may rest during certain times of the day. We need to consider social behaviors which have an impact on the distribution activity of the animals, and their diurnal circle should be also considered. Okay, now most important is the establishment of the strip width. The easiest approach is simply a fixed strip width. Why do we need that? We need the strip width in order to calculate the sampled area and later refer the number of animals counted to that area in order to get a density. The sampled area is calculated by the length of the transect line times the strip width on both sides of the transect line. If the terrain is open, like in a grassland or a desert area, we can keep a fixed strip width throughout the aerial sample. However, this is hardly ever the case, and in such cases we have to adjust our strip widths in accordance with the habitat type. In denser habitats, a smaller strip width; in open habitat, wider strip width. That will lead us to a visibility profile of changing strip widths, which is pretty tiresome because it has to be established prior to the count, and the strip widths will change permanently with the change in vegetation density. So what can we do? The best way to overcome this problem is to establish mean sighting distances. So for each of our sightings, this is now for a road count, we will establish the distance, but it's also applicable to aerial counts. We will establish the distance between the transect line in a right angle to the position where the animal is. So at the end, we will have for each sighting one perpendicular distance. This is a method which was already developed in 1945 by Kelka, and he went a step ahead and he lumped now the distances into categories of, in this case, let's say 10 meters. So in the first 10 meters away from the transect line, sorry, here, this x-axis in this case is the transect line. This is a distance of 10 meters. And within this distance, he counted 10 animals. In the second category, 20 meters away, also 10 to 20 meters away from the transect line, he counted again 10 animals. In the next section, 20 to 30 meters away from the transect line, he had only 5 animals counted, then 4 and 3. And if we plot now the number of animals against the distance category from the vehicle or the transect line, we will have in our first category 10 animals, in the second category 10 animals, in the third one 5, 4, and then 3. So we can visually now determine a fall off point that is here at 20 meters, because here we have a steep decline, in the number of animals. However, this is not always the case. In this case, we would have a strip width of 20 meters at the fall off point. But in such a case, if the categories are unregularly distributed along the distance from the transect line, we can determine no fall off point. The same for a continuous decline like in A and B. So in order to overcome this problem, a software was developed, and this software is freely available on the net under this link, and the software is called Distance. The method applied here we refer to distance sampling. So the software requires a number of parameters, such as the transect line L, this is this line here, and the position of the observer Z. We also need to have a clue or establish the position of the individual, the animal, which is counted here X. And we need the sighting of flushing distance, which is R. Usually an animal is accounted well before the point of a perpendicular distance to the sighting. So we establish the length between the first sighting and the animal, and the angle between the flushing line and the transect line, which is theta, the sighting angle. From the angle and the flushing distance, we can calculate now the perpendicular distance, y, or the software is doing that. And having all these parameters, the software will eventually calculate the effective strip width, W, on either side, and this then multiplied by 2, and you get the effective strip width on both sides. Now, doing that, we will oversee some animals within our strip widths, and others are outside the sampled areas and are also not counted. Others within the transect are counted, here expressed by the red dots and the perpendicular distances. The dark spots represent either the overseen animals or those outside the strip widths. And from all these variables and the information on the animals, the software will calculate, as in Kelka's example, a curve, or it will first lump the sightings into categories of distance from the transect line. Here, in this case, it is 100 meter, every 100 meter a category. And in the first two, we have somehow equal numbers of animals, but then we have a fall off point. So even if this is unregular, this example, the software is able to determine the strip width and give an abundance estimate. From this abundance, from this estimated abundance, then we can calculate the density relating the counted number of animals to the total sampled area. Okay, this program is called Distance. The method is distance sampling, published here in a book by Bookland and their colleagues. Distance sampling, estimating abundance of biological population, published by Oxford University Press. The program, the software, freely available under this link. However, some assumptions need to be applied when we employ distance sampling. All animals which are on the transect line, so 0 distance from the transect line, must be detected. It must be ensured that all animals standing on the road have been detected or were seen. The animals should be evenly distributed throughout the surveyed area. This is often very difficult to achieve. And animals should not move before first detection. Moreover, all measurements, the angles and the distances must be exact. So ideally measured by a rangefinder, using a rangefinder. Okay, this is how we carry out distance sampling nowadays using this software, and which gives us the effective strip width, the abundance of animals in the count area, and eventually the density, relating the abundance to the surveyed area. Now the question is, how often we actually how often do we need to count? Or in other words, for how many hours do we need to fly? Or how many transects do we need to count in order to get a reasonable, robust estimation of the number of animals in the study area? So on the x-axis, we have here now the confidence interval as a proportion of the counted number of animals. And this confidence interval decreases with increasing number of flight hours or with increasing number of transects. Why is that the case? As more often we count, as more precise is our estimate. That means the confidence interval gets smaller. So now from that, we want to establish a threshold, a maximum sampling efficiency at which we can stop to sample, to keep on counting. And you see here in the lower part of the curve, the change in the confidence interval is almost rejectable. It is almost not existing. So in the moment where the curve starts leveling down, we have the ideal confidence interval here around 20, where we could stop adding further transect or adding further flight hours. So in this case, around 20, 20 hours flying, I would stop my service and I will get a robust estimate of the animals in the study area. Good. Now let's think about non-invasive sampling methods. These count methods, which I have shown you so far, require that the observer is present in the field and that the animals are more or less disturbed by the plane, by the helicopter or the car, or even worse, when carrying out a drive count. So what methods can we apply to not disturb the animal? This we call non-invasive sampling methods. Or in many cases, the animals live in such dense vegetation or in inaccessible areas that these distant counting methods are inappropriate. So we can use various trails, tracks, and traces to estimate the presence and maybe even abundance of a certain species in our area. That includes tracks, droppings, localized identification sites of droppings. It also involves bones, skulls, and antlers, which were thrown off. Broken vegetation indicates that animals have passed, and bite traces on the branches give us also a clue that a certain species is present in the area. However, what are the constraints? We need to know the rate of defecation of the target species. How often do they defecate in order to make a meaningful statement regarding the abundance of the animal? Pellets and piles of pellets are often difficult to detect among the leaf litter, here in this example, or in the vegetation. And it is important to accurately identify the species and the age of the pellets, which is often difficult. You need a very profound knowledge on the shape, size, and consistence of the fecal droppings and the differences between species. And we need to know the decay rate, because after a few days, the pellets will shrink, they will lose water, and therefore change consistence, color, and shape. So this is often difficult and is only employed if no direct sampling methods can be employed. Good. Now to the second part of this presentation, to the application of trail cams or camera traps. Only in recent years, the technology advanced so much that this technology became widely available to conservation managers, and it becomes more and more sophisticated, provides better images with higher resolution, and different technical features which make this technology really, really useful when monitoring or counting wildlife populations. So camera traps are the most patient and focused field workers you will ever have, willing to quietly sit in one spot, waiting for a rare event to unfold in front of them. Imagine you would to have to carry out this work by observers. We would have fatigue, exhaust, and the bias of the observers. So waiting for these rare events. Rare events are termed long tail events, such as the encounter of a predator, or this example here, anteater deer in the canopy of the tropical rainforest in South America. The camera was installed up in the highest trees of the forest and caught this very rare picture of an anteater bear mama with their baby carrying it on the back. Camera traps are very non-invasive, not like our distance count, because the cameras are well camouflaged. They do not emit light or sound, and therefore the animals hardly ever detect them. And they allow the recording of relatively natural behavior because the animals are not disturbed. Good. So what are the advantages and disadvantages of camera traps? First of all, minimal labor costs. Nobody needs to sit there and wait for the animal with the binoculars or a tally counter. The camera will patiently wait and has only the equipment costs, which can be also pretty high at times. As I mentioned already, camera trapping is non-invasive, but it cannot obtain physical samples like hairs for DNA analysis. Camera traps work 24 hours throughout without interruption unless there are technical problems. The disadvantage is that we can sample only endothermic species, those warm-blooded animals which emit heat. Camera traps usually react to movement and to heat. And if we have ectothermic species like reptiles or amphibians, camera trapping technology cannot be applied. We get a wealth of other data, not only the abundance or presence-absence data, but also on the behavior, the body condition of the animals, their activity patterns, and even on the local habitat conditions. However, often it is not possible to identify individuals. We will come back to that in a minute. I mentioned this already. Camera traps have no bias. Human observers are often biased towards certain expectations. They get tired or distracted. So camera traps are patiently waiting for the event. Camera traps, on the other hand, are sometimes malfunctioning. You may forget to change the batteries, or the sensors are not properly working. The flash is malfunctional. So there is a risk of losing data due to malfunction. Camera trapping images can be used as digital specimens and saved in museums as Wuxia specimens, and they are easily to be catalogued. However, there are legal and ethical risks. People can complain because a certain area is surveyed by camera traps. So legally, you should put a warning signpost in order to inform people that this area is under CCTV. observation. And of course, theft and vandalism need to be considered when placing camera traps in the wild. So how to design such a camera trapping study, and what data do we derive, and what can we obtain from those data? What information we can obtain? Here an example from Indonesia, Borneo, one of the islands with a protected area in the northwest. This is the protected area, and we want to know what animals, what is the species richness of the study area. So we establish a camera trapping grid, and comprising of several squares, in which center we establish our camera trap. So one camera trap in each grid cell, from which we can then extrapolate to the area of each grid cell. So the camera is somehow centrally placed within each quadrant, and we will get from each side, from each square, now plenty of pictures on every day of sampling. That will result in plenty of images, and the data, the accompanying data will be summarized in Excel tables, from which we can start then doing our analysis. So what parameters can be established from such a camera trapping study? For sure, the activity patterns, whether the animals are more active during the day or during the night. We can establish the species richness, which is simply the number of species present in the study area. We can establish the relative abundance. The relative abundance is the number of events per camera trap per unit sampling effort for each species. And most important, we get information on the distribution of animals expressed as the occupancy. The occupancy is the number of camera trapping locations where a species was detected, divided by the total number of operational camera trapping locations. Good. Here in summary, once more in this table, what are the specific topics that can be investigated using trail cams? This is the absence or presence of a certain species in a certain area. It is the species distribution within that area. I just described you, the expressed as the occupancy. We can establish the relative abundance, the species richness, but also habitat use, activity patterns, phenology, foraging and feeding ecology of the target species. We can make statements regarding predators or frugivory and mutualism even. And with regard to humans, we can make statements regarding illegal hunting, poaching, crop raiding, or the invasion of livestock into a protected area. This is basically the same information presented as a flow chart. We can use camera trapping technology to establish the presence or absence of species, the diversity under certain circumstances only if we consider richness. This is what we will look at in detail in the next two slides. We can establish abundance, but only if we know individuals, and that can be achieved by a capture-recapture design. We can establish distributions expressed as occupancy. We can measure certain behaviors such as activity, and we can make statements regarding the people, poachers, crop raiding, or domestic livestock invading a protected area. So let's have a look at these two examples from the Boni Dodori Forest Reserve in coastal Kenya, where we were looking, or where ZSL was looking for the Aedes duiker and the establishment of species richness of the larger ungulate species in Parc National de Haut Niger in Niger. So this is the known distribution of Aedes dica in East Africa, only on the island of Zanzibar, but scientists suspected that in Boni Dodori and Arabuko Sokoke forest in coastal Kenya, there might also still some of these species have survived. So a cameras trapping study was implemented in 2012, and it discovered indeed a population of the critically endangered Aedes dica. So this was a presence-absence study, and we simply confirmed the presence of this endangered ungulate species in the two target areas. Another interesting thing which was discovered during that survey was a yet undescribed elephant shrew. And the second example I told you was to establish species richness. Here in this case, we wanted to know what different species of larger ungulates and their predators occur in the sample in the study area. So we established species richness, the number of species encountered, and we established the observed species richness, but from that, using technology, statistical approaches, you can also estimate the estimated or true species richness, because you will never observe all species by through the camera traps. So that allows us to make statements regarding the richness, but not to establish diversity indices like Shannon index or the evenness index. That would require that we can distinguish individuals, for example, by their individual fur pattern. Sorry, so back to our flow chart. Now I would like to draw your attention towards establishing the relative abundance, which is that what we usually want. We want to know how many animals of each species are roughly there. So one measure to express that is the relative abundance, but that can be only achieved when individuals can be distinguished individual, therefore avoid double counts. And that is usually achieved through the application of a capture, mark, recapture scheme. So what is that? First of all, we need to make sure that we can individually distinguish our animals. Here, for example, the bushbok again with its characteristic harness pattern, the stripes and dots, which are different on both sides of the animal and which are individual, like our fingerprints. So having camera trapping images from both sides will allow you to definitely say that is this in this individual. If that is not existing, we need to capture the animals in order to mark them, for example, with ear tags, or in the case of the butterfly by some color dots. And this will enable us now to employ capture, mark, recapture methods. How does that work? Let's say a pond is our protected area, and we want to know how many fish individuals live in that pond. What is the relative abundance or the density of fish in the pond? So we start fishing and we catch 10 fish on the first day. And we mark the fish, like with color dots here on the butterfly. And all the 10 marked animals are now released back into the pond. Now, on the second day, we will use the same method, the same pond, and spend the same time fishing again. And we will catch another 10 fish. This time, 5 of them will have a marking already from our first catch, but the other 5 have no mark. Now, from the ratio between marked and unmarked animals, we can estimate or calculate the estimated population of fish within the pond. So we take the number of tagged, also marked fish on the first day, it was 10, divided by the percentage of marked fish in the second sample. So half of them, 50%, expressed as a rate, 0.5, was found on the third day. So 10 divided by 0.5 makes 20. So our estimated population size is 20 fish inside the pond. Now you can do that again and again and get more and more precise estimates as longer as you recapture the marked individuals. Here another example of a species which has natural markings. The tigers have individual stripe patterns on both sides, and establishing now the grid, the camera trapping grid with the squares, one camera in each square, we can get very detailed information about the movement patterns and the habitat choice of the tigers, for example, their home range size. So our cameras will spit out again the Excel sheets, and we can relate that to the total study area and get the conventional recapture rate, or we make it more spatially explicit, relating our data to different habitats in each square, so we can transfer this information to the map of the study area, put the tiger observations in relation to the habitat types, also here expressed as whisker box plots. Good. Now it is not always possible that animals have markings, and many have markings in invisible areas of the body. For example, this here is a wolverine marten, a kind of mongoose from our boreal forests here in the north, and they are looking all the same, except that they have very individual patches on the ventral side of their neck. So these researchers established a construction where the wolverine has to climb, and they put a bait so that the wolverine has to stretch its head upwards to reach out for the bait. And this is a moment where the camera can capture the patches on the ventral side of his neck. Very interesting construction. So often these characteristic marks are not visible, but with some innovation, you can make them accessible. Now, as I told you about the advantages and disadvantages of camera trapping technology, I mentioned that often no physical contact to the animal can be obtained, and it is therefore impossible to get hair samples or skin samples or blood samples. So DNA analysis are not possible. The DNA cannot be related to the focal animal, and that's why this research group invented alligator clip snaps, which are placed openly on the place where the wolverine has to climb, and once it touches them, they snap and collect some hairs for DNA analysis. That has now a big advantage. We can make statements not only about the distribution and abundance of the animal, but also their genetic setup within the population. For example, the relatedness between single individuals. Okay, I would like to terminate my lecture with some recommended reading. This lecture today was largely based on publications by the WWF Counting Wildlife Manual, or the Camera Trapping Manual by WWF, and also very recommendable the Handbook for Wildlife Monitoring using camera traps. Here are the references, and I say thank you very much for your patience and for your interest, and I want to say goodbye with one of Ethiopia's most enigmatic animals, most endangered antelope species, the Swainey's hartebeest from Zinkele, Alcelaphus bucelaphus swainey. Thank you very much, and bye-bye. See you next week again, I hope, and then I will report to you about wildlife reintroductions and translocations. Okay, cheers from Liverpool. See you next week. Bye bye. See you.